On behalf of myself, Theone, my name's Theone. I'm the local site principal investigator of the CLSA for Victoria, and Tarek, who's in the audience somewhere, is my local site principal investigator. They recently renamed us, so it's a mouthful. But on behalf of both of us, I'd like to welcome you guys here. And sorry, I'm having a screen issue for some reason now. There we go. Welcome you to this forum to showcase some of the work that's being done with the CLSA data by local researchers. I'm not getting a slide advance here. Maybe I do need to use this, hey? Okay, let's try that. Nope. Oh, there we go. Okay. Okay, so I'd like to start us off with a territorial acknowledgement. So for all of us present at UVic today, we acknowledge and respect the Lekwungen, the Songhees, and Esquimalt peoples on whose territory the university stands, and the Lekwungen and Wasanich peoples on whose historical relationships with the university and the land continue to this day. I was going to say that it's a lovely day here in Victoria, but it's kind of not. But I hope that everybody online is having a lovely day wherever they might be. It's a little bit messy and rainy here. But I'm sure we have many lovely spring days to come, hopefully. So, all right, let's continue on. Okay, so this is just a quick overview slide. I'm going to do a very short intro to the event as a little bit of a refresher about the CLSA for us all. Quickly discuss the CLSA main study and the sub-studies a little bit to try and just get that organized in our heads before I present hand over things to today's presenters. So just to start things off, this is a picture that was recently taken at a CLSA operations committee meeting, our first in-person meeting since the pandemic, which was nice last summer. Not everybody's in this picture, but you can get a sense that it's a pretty large team. If this is just the people involved in study governance, and then there's everybody who works at all of the sites and all of the researchers as well that work with all of the data once you guys, with your lovely data-giving personalities, submit all of your stuff through the CLSA portals and all of that. So moving on. So I know that you're all familiar with the CLSA as participants, and I think we have some family members online as well. So I'm not going to spend too much time reviewing what the CLSA intends to do, but in a few words, the CLSA at this point is a research platform with the aim of providing the infrastructure to enable state-of-the-art interdisciplinary population-based research and evidence-based decision-making that will lead to better health and quality of life for Canadians. So findings from the CLSA research platform aim to identify ways to prevent disease and help improve health services, to examine how non-medical factors like economic stability, social change will influence people as they age, to answer questions relevant to decision-making in health policy and government programs and services, and to determine how biological, clinical, psychological, and social factors influence disease, health, and well-being. And also as a byproduct of the study being run to train future generations of researchers as well, who will use the CLSA data to examine other areas of research on aging. Oh, keep trying to press the button on the computer instead of this button. So this is just a slide to indicate the scope, which, as I'm sure all of you are aware, there are 11 data collection sites in provinces across the country. And this is a very large study, so not just local to Victoria, but really everywhere across Canada. And so among the approximately 51,000 participants in the study from across Canada, data are collected from over 21,000 people using telephone interviews, which we refer to as the tracking cohort of the study. So you may be part of that cohort. The remaining just over 30,000 participants provide data through in-home interviews and data collection site visits, referred to as the comprehensive cohort. And so many of you may be part of that cohort as well. And as you can see on this slide, the main study completed baseline assessments in about 2015. So many of you have been with us from somewhere between 2011 and 2015. Follow-up 1 assessments completed in 2018, follow-up 2 in 2021, With, by the way, I found out when I was looking stuff up for this study that 96% of people completed follow-up to assessments. So that's a testament to all of you and your diligence in completing these assessments, which is just wonderful. That kind of statistic for a longitudinal study is relatively unheard of. So the CLSA has some fantastic participants. And then right now we're in the middle of, well, mostly done, I guess, completing follow-up 3. We're hoping to be finished in the fall so we can move on and cycle up to follow-up 4. So many of you may have already completed follow-up 3 assessments as well. And then we've got, as you can see on this slide, after follow-up 3 is done, we have 3 more scheduled follow-ups over the next 10 years. So we will be reaching out to you very soon again if we haven't in a couple of years. Okay, and so as you're all aware, because you provide the information that the CLSA collects, we collect a lot of data from questionnaires, either in person at the collection site, in home, or over the phone. And this includes demographics, details about many aspects of your lifestyle and behavior, social life, work, physical activities and health, psychological health, medications, thinking abilities, cognition. And then you're also participating in physical assessments. There's biospecimen collection, blood samples, and so on and so forth. And then you also complete a bunch of cognitive assessments, which helps us get at your memory, your thinking abilities as well. And then recently with follow-up 3, the CLSA has also added some new questionnaires. So many of you, if you've already participated in follow-up 3, would have had some of these. And others, if you haven't yet participated in follow-up 3, this might be what you have to look forward to. So questionnaires including data on quality of life, sleep, nutrition, healthcare utilization, sexual health, and family history of diseases, and then some new assessments of vision and olfactory functioning, and some new biomarker collection as well as repeating some of the previous biomarkers, epigenetics, metabolomics, and things like that. I'm getting to that. So here on this next slide, we have one of our wearables. So in 2002, the CLSA also introduced some wearable activity trackers for some groups of participants. So not everybody will do these, but some people will. And the idea is to get new information on sleep quality, and it basically kind of monitors how your brain activity is while you sleep. This one does this. You can see he's wearing it on his head there. And then we also have some other ones that are meant to monitor your actual activity when you're awake. So how active or sedentary a person is, as well as looking at community mobility, driving, and how people utilize things in their communities. And so these are just devices that you would wear on your wrist, or for the sleep one on your forehead during the day or night. Okay. And then this slide here just shows some of the additional sub-studies you can see coming in starting in around 2020. Some of you may have participated, many of you actually may have participated in some of these. And so I'm just going to very quickly go through these sub-studies so that you have a sense of what each one was, and kind of map them out a little bit for you. So we'll start off with, there were 3 different COVID-related CLSA studies. The first was a questionnaire study, which was launched in April 2020, so soon after the beginning of the pandemic. And this collected data from almost 30,000 CLSA participants, and this was through online and telephone interviews. These were, I think, over about an 8-month period. And the study explored the effects of the pandemic on older adults, including looking at coping, impacts on physical and mental health, and changes in how older adults access healthcare services during that time. There was also a seroprevalence study, and this one was looking at blood samples of about 19,000 CLSA participants. For this study, blood samples were collected either in person at a data collection site or through a finger prick kit that you would have done at home and then mailed back in. And the aims of that study were to estimate levels of immunity among older adults and to aid in understanding how COVID-19 infection was affecting people. And then the third of these studies, which is actually a longer-term study that's still going on, is the COVID-19 brain health study. And this one aimed to examine how COVID-19 affects our cognition and the brain, both in the short and the long term. So for this study, a subset of participants completed a phone interview and may complete some magnetic resonance imaging. at a local clinic. And the focus of this study is understanding possible covert longer-term effects of COVID-19 on our brains. The people for the last one would have been people that had COVID. They may, you know what, I'm not 100% sure because they may have also been looking at a control group. So people that they could confirm hadn't had COVID at the time as well. Yeah. Sorry? It could be. So they started this before that was the case, but I think this would be to look at maybe who might become a long hauler and who might not. So I think they were probably started assessments in a shorter period after infection that would be considered long haul. But they are looking in the long term as well. So they're following up with people. And so my guess is that some of the people in the study probably would kind of be those long haulers, and others people may have already recovered. Yeah. Okay. And then there's a couple of other studies that are not COVID related. So another one is the Weston Healthy Brains, Healthy Aging Initiative, which you may have heard a bit about if you've looked on the website at all recently. I know they have some showcase materials about it on there. This study uses brain imaging and gut microbiome analysis via stool samples to examine mechanisms that influence brain health and healthy aging. And it'll include a subset of 6,000 CLSA participants when it's all done. All 6,000 will provide stool samples and use activity and sleep trackers. And 2,600 of these participants will also complete MRIs and undergo some additional cognitive testing above and beyond the normal testing in the CLSA. And the aim for this data is to inform the development of screening tests and prevention strategies for brain health and aging. And then last but not least, we have the CLSA memory study that's just completed its data collection. So for this study, 600 participants with a range of cognitive abilities completed a 1-hour medical assessment with a study clinician. They provided some additional medical history, completed a brief cognitive screening test, and a neurocognitive evaluation. And for each of these participants, a family member or friend also completed an interview over the phone about the participants' cognitive health, their abilities to complete daily tasks, and their behaviors. And the goal of this study is to examine individuals with and without memory problems and to see if the medical assessment outcome indicating whether they have a memory concern or not could be correctly predicted using data already being collected by the main CLSA study. So the goal really to see if there's a possibility to use the normal CLSA main study data to create an algorithm that would predict some memory changes or cognitive problems for somebody. Okay, and so overall, through participation in the main CLSA study, and for the many of you that have probably had a hand in some of these sub-studies as well, the CLSA has collected a lot of data. As you can see, we've gone through a whole bunch of things, and you all have contributed significantly to that, which is very important. I also want to mention that there's some other data that's now can be linked to your individual data with the CLSA via connection of your CLSA data within the main data sets with environmental and community-based data. And this is data that's collected by postal code or region. So this includes things relating to the greenness of communities, weather, air quality, noise, light, employment access, and other related information that can be examined together with your CLSA data based on your postal code and things along those lines, which is super interesting and also just extends the utility of the data that you're able to provide. And so with all of your contributions and all of this data that has been and is continuing to be collected, CLSA is rapidly becoming the most comprehensive research platform for health and aging globally. At this point, we have over 500 research teams and stakeholders using CLSA data. There have been over 350 approved project publications using this data. And the CLSA has contributed to development and training of 500 or more, probably more at this point, highly qualified personnel since 2011. And here are just some of the many connections, funders, and partners associated with the CLSA and developments and collaborations that relate to this research. Overall, there's a vast network of partners and contributors. And most important contributions are, the most important contributors to all of this are all of you, the participants, because without you, we wouldn't be able to have any of this data or be able to speak to any of these very important questions in healthy aging in Canadians. So that brings us to why we're here today, which is to share with you some of the awesome research that is being done locally right now using the data that you've contributed to the CLSA platform. So all of our speakers today are affiliated with the Institute on Aging and Lifelong Health at the University of Victoria. I'll introduce them a little bit more fully as they come up, but we'll hear from 5 presenters today, all of which whose beautiful pictures are on this slide. First up, we're going to start with Dr. Stephanie Calce, and just a very brief bio. Dr. Calce is an adjunct assistant professor in the Department of Anthropology. She is a bioarchaeologist who studies age variation in the human skeleton, senior lab instructor in the Department of Anthropology. Welcome you to come up.
Okay. Welcome and thank you so much. Yes, my name is Dr. Stephanie Calce. It's okay, it's hard to pronounce. And I'm from Anthropology. So this is going to be perhaps a little bit of a departure in terms of the content, the research sort of trajectory that you might be used to hearing about in the context of this data and research. Because I'm a bioarchaeologist, I'm going to look at osteoarthritis. Oh, that's okay, I'll move it. I'm going to consider osteoarthritis and bone mineral density in the past. So we're really thinking about this in the context of archaeology, which is quite different probably from what you're used to hearing about in terms of health-related data. Okay, let's see if this will work. Nope, not that one. Oh, hello. Great. So just a quick outline of what I'm going to cover today. First, I'm going to tell you about what biological anthropology is, because some people know, a lot of people don't. It's biology, it's anthropology. What does it mean? We're going to think about the problem of OA in the past and present. I'll tell you my research questions, some of the methods we've used, and how the CLSA data set is helping us to answer those questions, the results, the conclusions, and most importantly, where we're going next, because there are lots of things still happening. Okay. So biological anthropology, what is it? What do we study? It turns out quite a lot of different things that are seemingly unrelated. Things like modern human biology, there's the paleo environment, fossil hominins, living primates, and all of these sort of things seem very disparate to each other. But in fact, what unites us as biological anthropologists is the study of human variation. And so that's what we're all interested in. And some of the questions we ask as biological anthropologists are, why do we vary, right? What are the benefits and consequences of that variation for the success of our species? And I think what's the most important or interesting part of what we study is where we find this information. Because in fact, all of human variation exists on only 0.1% of our genome. So that's kind of the coolest part of all the questions that we ask. The answers are really existing on a super small part of our shared biology. And lots of things we can study. Why study the past? Very good question. Basically, we try to map evolutionary and life histories. We're thinking about antiquities of disease and using all of that to understand the process of biology. In my case, I'm interested in osteoarthritis and bone strength and thinking about that in today's populations, right? To help us learn about health and lifestyle of archaeological past. And we're interested in lots of different things, how and why osteoarthritis develops at different ages. We're interested in how different activities can affect bone health patterns. And so combining what we know about osteoarthritis and bone mineral density today with what we find in ancient skeletons actually helps us to paint a much clearer picture of how people lived long ago, which again is important to understanding things like aging, reproduction, and other life history characteristics. For me specifically, I'm really interested in health-related outcomes that are to do with age variation in the skeleton. So what is it that we do that have an effect on our biological longevity, not just living long, but living well? And another outcome of my research also maps the process of bone aging across the adult life course. So how bones change with age. And as a bioarchaeologist, also a forensic anthropologist, we can use that information to estimate age at death from the human skeleton. So we can actually tell how old somebody was when they died. Okay, so what do we know? Lots of things. Bone, first of all, I should tell you, changes throughout our lifetime. Okay, so when we're growing, we put on lots of bone. When we stop growing, we call that skeletal maturity. A different process takes over to help with bone maintenance for the rest of our lives as adults. This process is called bone remodeling. It's a relationship between forming and resorbing bone. It's a continuous process, but the rate of this process actually varies for every single person in the room. So this is a unique variable. And while some may form bone at a uniquely rapid rate, others may lose it. You might have heard that before as osteoporosis. And this varied speed with which we build, we resorb bone, we call this the rate of aging. And as researchers, we want to know, well, what are the things that people do in their adult lives that affect this process? So what would be an optimum threshold, and how can we maintain good bone quality well into our later decades so we can, continue to move around well and often? Here on this slide, I've just given you a brief outline of the kinds of things that we study in the archaeology of osteoarthritis, so questions around aging. This question is how our physiological age matches with our chronological age, so how old you look is actually overlapping with how old you are, right? We want to know if people in the past were engaging in more or less strenuous or repetitive tasks and what that means for our bone health. And then finally, things like dietary habits and nutritional status. Can we use the kinds of things like diet and nutrition to look at deficiencies and how those reflect in things like our bone mineral density? So we've studied osteoarthritis for a really, really long time. And it's long been understood as a mechanical problem. It's sort of an issue of wear and tear. It's this idea of a disease of joints. But we also know it is related to aging. And therein kind of lies this conundrum, because if osteoarthritis is a disease, then it should actually only affect a really small proportion of the population. But if it's related to a necessary part of aging, well, then you kind of think everybody's going to have it the older they get, right? And what became clear when I kind of started to consider these OA definitions was that even after more than 75 years of study, the mechanisms that underlie OA progression are relatively misunderstood. And not surprisingly so, because people like me and also clinicians are only ever dealing with half of the information. And what I mean by that is, as a paleopathologist, I have the bone itself, right? But I don't have any patient history. We can't go back and ask individuals, what they were doing and look into their daily lives much more clearly. And clinicians actually have a lot of information about behaviors, right? They have the information of the life history, but they don't have the joint surface characteristics. And in fact, their process of identifying clear onset and progression of OA is related mostly to x-rays that are hard to read, or pain, which actually doesn't correlate at all with x-ray. So because pain doesn't map, or sorry, x-rays don't map onto pain experience, we're both dealing with sort of a disconnect in asking those questions. So marrying these two fields is actually going to be really important to tell the full story of cartilage degeneration, subchondral bone remodeling, and joint degeneration. Using the CLSA dataset is critical, actually, in being able to answer those questions. Because, as I said before, my goal is to accurately age the skeleton at the time of death. And if we don't understand the process of aging, well, then we have to wonder if our methods are doing a good job. Okay. One sec. That's fine. Okay, so osteoarthritis and bone mineral density are linked, and we think that it is related to the rate of aging. And so we're asking a question, a very simple question, to sort of get at one of these answers. Is bone mineral density relatively higher among people clinically diagnosed with osteoarthritis? Okay, and this is a significant question to ask, because when we look to the literature, we actually see a lot of opposing results. There are many ongoing studies that show, yes, bone mineral density is higher in people with osteoarthritis. They call them like super bone formers. We see also another part of the research that says no, less bone mineral density, lots of bone loss. And then we have even more people saying, no, it's a variable relationship. So there really is no consensus in the literature about answering this question. And what that tells me is that we're not quite done investigating the relationship between bone mineral density and osteoarthritis just yet. And it's really no wonder why we can't replicate these findings in an archaeological setting where we have much, much smaller, incomplete data sets. Okay, so how did we do it? So again, I'm interested in investigating the variability in relationships of bone density and osteoarthritis, and we're going to do that by sex, by age, and activity, and looking at it in a couple of different ways. So we're thinking about the rate of skeletal aging in the context of high and low bone density forms. So where do people fall on this spectrum of bone remodeling? We're thinking about the lifestyle factors that may be influencing that relationship of adult bone metabolism, what things speed up, what things slow down. And then finally, to improve our methods of identifying osteoarthritis and osteoporosis as a consequence in an archaeological sample, as well as to estimate age at death. Okay. So some of our preliminary results, again, this is just an ongoing study. It's just a snapshot, a peek into what we found so far. And good news, we found, yes, bone mineral density is relatively higher among people clinically diagnosed with osteoarthritis. And we found that significant across all the groups in our sample. So we looked at everyone, we separated males out, we looked at females, we looked at different age cohorts, we looked at that by males and females as well. And we also separated menopausal females because there's a lot of hormonal influence on bone quality as well. But what we found is that, yes, increased bone mineral density is a risk factor for osteoarthritis. Okay. Pardon? That's correct. Yep. And that's leading us to an interesting result, but also just more questions, to be honest. We've got a lot more things to study to sort of tease apart that relationship and that understanding a bit more. We're interested in looking at lots of different sites of bone mineral density. So you can imagine parts of your body that you put more load on, right, versus not. So things like your shoulder joints, your hands, your wrists versus like your hips and your knees. Maybe there's a different relationship there. We're thinking about also the measure of activity. So strength and exercise, how does that matter? You're probably participating, those of you at the data collection sites are doing grip strength things, right? They're making you stand up out of chairs, they're making you walk around. It's really encouraging to hear that there are mobile devices being added because we're interested as well in like physical activity and literal physical activity like walking on a regular basis and other things that may be considered strenuous. So that's a really exciting data point. We're really interested in epigenetics as well. My co-investigator, who's not here in person, but maybe is online, hi Allison, hi Annie, is looking at epigenetics and metabolic syndrome, in particular how oxidative stress relates to osteoarthritis and aging. And then finally, we're really interested in all of these changes through time. So for us, the CLSA data set is super important because again, as we studied archaeology, a one point in time, right? We're looking at the point of someone's death. So it's not it's a cross-sectional type of study. We don't have this information in longitudinal form. We don't know how that changes through time. So we're really interested, and that's super unusual for us as archaeologists to be able to study that relationship. Yeah. Oh, it's a longer conversation, but essentially it's the way in which your genes are turned on or off. So yeah, what parts of your genetic blueprint are being used in any one particular way. And we're interested in epigenetic acceleration of aging. So this is a question about how old you are mapping onto how old you look. So if you're epigenetic aging faster, what does that tell us? If you're epigenetically aging slower, what does that say? Right? How can we tease that apart? Yeah, so that has to do with the rate of aging as well. Okay, well, that's all I want to say about this, but mostly just to say thank you so much for participating, because again, we really can't ask these questions in a meaningful way without this type of data. I'd like to acknowledge that our research is supported by an Insight Development Grant, and also our undergraduate research assistant is also here. We have many other people involved in our project, the Phase Research Group, the Gab Lab, and all of our friends at UVic. Thank you so much.
Okay, so I'm first going to introduce a term that's called mild cognitive impairment. So it's kind of thought of as, it can be like a phase before dementia sets in, but also some people can stay consistently in that mild cognitive impairment stage. But really what it is, there we go. Okay, so it's really when there's objective evidence of cognitive impairment via cognitive testing. So what we mean by objective evidence is you go in and you might do some paper and pencil tests, answer some questions, and then when we look at those results, we see that compared to people of the same age and the same education level, the scores are lower than we would expect. But there's also the fact that these deficits don't really impact day-to-day. So in dementia, these deficits are a lot more severe, and you see that impact in terms of how someone's able to function. In cognitive impairment, the deficits are milder, and they also generally don't impact someone day-to-day. So it's believed that the prevalence is about 15 to 20% of people over the age of 65. And it's also been found that 25% of people who meet criteria for MCI, who are classified that way, will actually revert to normal cognition. So there's groups of people who have this diagnosis and then are actually okay, versus other people who will then go on to develop dementia. So this is a little bit of a graph form of that. So in the green arrow at the top, you see normal aging. So the y-axis there is cognitive function. And in normal aging, we do expect a little bit of decline over time. That's normal as we age. And in dementia, you see that drop off happen. So where the arrow is orange there, you have that MCI range. So you start to see that decline in cognitive function, and then potentially for some people that turning into dementia. But you also see those dotted lines there. So you see the one that some people actually revert to normal state, and some people stay in that stable MCI state. So still having some cognitive issues, but not really affecting them day to day. So it's a lot less impactful in their life than the people who go on to develop dementia. Okay, so we wanted to focus a little bit on that MCI group. Why are some people going on to develop dementia? Why are some people staying stable? What are the factors affecting that? So there's a lot of research on this. There's a lot of risk factors associated with that. Things like hypertension, things like diabetes. But there's also these kind of more protective lifestyle types of factors, like physical activity, like social connection. So we're a little bit interested in looking at those. Those are ones that are a little bit easier for people to modify, right? To increase exercise, to increase social connection. So we're interested if those changes could make a difference over time. So the first study aim was, how do physical activity and sociality, so how social people are, what kind of social involvement they have, how does that affect cognitive function, so things like thinking and memory, over time in older adults who are experiencing cognitive problems. So already people who are starting to notice those changes, can changes in those lifestyle factors, in physical activity and social function actually make a difference over time? Here we're kind of saying cognitive problems because in the CLSA, we don't have an MCI category. We don't have people classified as mild cognitive impairment. So we're looking at people, we're looking at their scores on those tests that we administered, and we're saying, okay, are these people performing lower than we expected? And we're sort of defining that as cognitive problems. Okay, and how do these risk factors affect cognitive function over time? So if someone, for example, at the baseline were to report a certain level of physical activity and then they actually show a big increase over time, is that making an impact in their cognitive function? I also wrote a note here that we did, for some of these aims, we had some researcher partner involvement. So we had people who were caregivers of people with dementia helping us with some of these study aims and research questions. This was actually for a similar study we did with another large data set, the UK Biobank, but we did use some of the insights that they gave us on that data to translate to some of these CLSA questions as well. Okay, so obviously this is the CLSA timeline that we just talked about. So I just wanted to mention that here we're kind of just looking at baseline and follow-up one. is when we requested the data, this was what was available. So we're just looking at the baseline 2010 to 2015, and then the first follow-up collected between 2015 and 2018. Okay, so how are we defining cognitive problems? So as I said, it's those people who are falling below where we would expect. So in the dark blue there, that's kind of people who are scoring in the average range, would fall somewhere around there. And then people who are scoring 1 standard deviation below, they're scoring towards the end of that graph there, less than what we'd expect. So that's a clue that they're having some cognitive issues. And we don't look at that just for a single test. We look at people who are scoring that consistently for a series of tests. And so this applies to people who scored low on some of these different tests. I'm not going to go into too much detail here, but some of the tests are things like memory. So things like learning lists and then having to repeat them, things like executive function. So that's having to keep track of things, having to keep track of numbers and then letters and switch between them, and things like verbal fluency. So being given a letter and having to name as many things that start with that letter as possible, things like that. So people who are scoring lower on those tests, on a series of those types of tests, that's what we're looking at and kind of defining there as those cognitive problems. The other criterion is people who are showing decline over time. So not just people who at one time point are kind of showing these lower scores, but also we're seeing that decline over time. So I won't get into this too much technically, but there's something called the reliable change index, and it just allows us to know, okay, is this a change that is actually reliable in terms of not just a fluke, something that we can consistently rely on as a change. Okay, so in terms of the participants, just to give you a summary, people with cognitive problems we're defining as those people who scored lower and showed decline at the follow-up time point. And we're excluding people who already have a diagnosed neurological condition or event, so people who are diagnosed with MS or who've had a stroke. And our final sample size was 196. So this is really with a small subsample because most people aren't fitting into this category. Okay, so the variables of interest here, we're looking at performance on the 6-test cognitive battery, so all those cognitive tests at baseline compared to follow-up. And then we're looking at predictors. We're looking at what are the changes over time, as well as physical activity, and then a couple social variables that I want to explain a little bit what those are, and looking at how those potentially changed over time. So the physical activity one is based on this questionnaire. It's self-reported physical activity, and it gets at many different types. So not just going to the gym, but things like occupational, household, and leisure activities over the previous 7-day period. Then the social ones, we looked at a couple. First was social support availability. So that's a bunch of different areas of social support. So how do people rate the availability they have in terms of emotional support, in terms of positive social interaction? The next one was social participation. So people reported how often they were engaging in community-related activities. So this could be things like club, sport, neighborhood programming at least once a week. This was a categorical variable, so we just sort of had to choose what frequency we were looking at. So we chose once a week for this one. And then the last one was number of people you consider close friends, excluding family members. So people would just report a number. And then these are just some basic things we controlled for. So things like age and sex and ethnicity. Okay. And then we also did control for depression, as that has been shown to have an effect on cognition. Okay. So all these control variables were collected at baseline, and we had depression scores from both baseline and follow-up. So I won't go over the demographics too much, just to say we had average age at baseline of 66 and about 69 at follow-up. And in terms of employment, most people were employed. Pretty low depression scores, baseline and follow-up. Seeing some decreases in that physical activity score, and pretty good numbers though in terms of those social variables. Okay, so in terms of the findings, so the first model we ran told us that about 23% of why people's scores on that cognitive battery were different has to do with person-specific unique characteristics. So this means if we're looking at the whole group, we're looking at the decline over time, there's sort of those group factors and we see the decline, but there's these individual factors. People are different in terms of the amount of physical activity and the amount of social interaction they have. So this just tells us, okay, this is worthwhile to look at. There's something else going on here in terms of individual factors. So the second model we ran told us that the mean score for the cognitive battery was about 85. So that tells us it was about 1 standard deviation below average, which is what we expected because this is the sample we're looking at. And there was on average about a 4-point decrease in that score over time. And so the main findings, so this is where we added all the variables in. First, we were looking at if any of the control variables were significant. So we did find that white ethnicity was associated with higher scores at baseline. I'll talk about that in a little bit. But the more interesting finding is that an increase in the number of close non-family member friends between baseline and follow-ups, people reporting more close friends, was associated with more stable cognitive scores over time. So having that increase in that quality and quantity of that social interaction. So this is specifically, we're asking close friends, people you feel that you can confide in. People having an increase in that number, it did show more stable cognitive scores. We'd also noted some trends. These are things that are not significant, but we do have a small sample size, so it's possible these play a factor in a bigger sample. So we saw that higher depression scores were associated with lower cognitive scores at baseline. So people who are experiencing more of those symptoms of depression are going to score lower cognitively, which is consistent with some research out there. As well as that social support availability. When people talk about their emotional social support, that was associated with higher scores at baseline. So in terms of implications, there's sort of the thought that increasing one's social circle may be associated with that preserved function over time in people who are maybe more cognitively vulnerable or already starting to experience some of those issues. And so this implies that older adults who are having those issues may benefit from some of those lifestyle changes, increasing some of that social interaction to reduce that risk. So looking at that MCI group, when people are starting to experience that decline, there's maybe a possibility for some of those lifestyle changes to actually have either, be in that kind of category of stable MCI or potentially even that reversion to normal cognitive function. Lastly, I just want to talk about, so there was the factor that people with white ethnicity had the higher scores. Somewhat not surprising because our sample is very skewed in terms of having 95% people who are identifying as white. So when we're comparing the norms to people in that category, it's going to skew the scores somewhat. Depression and social support availability, although those weren't significant, those also might pose a protective factor as well. In terms of future directions, I am looking at including some genetic data in this because we know there's a genetic component to dementia to Alzheimer's. So there's a certain gene that we could include and see if that makes a difference in terms of what protective factors are more or less helpful. And other analyses, I might try to look at a bit of a bigger sample and even people who aren't experiencing cognitive problems, what effect are these physical and social variables having on cognitive function over time? And also including some more follow-up data just to see how things look over time. Yeah, otherwise I want to thank you, the participants, without whom this would not be possible, as well as the funders that were associated with this work.
All right, great. Okay, so hello, my name is Allison. I am here to talk about my master's thesis. It's titled Psychosocial Predictors of Cognition in Parkinsonism. So to start off with, I will define Parkinsonism. Most of you may have heard of Parkinson's disease, but Parkinsonism specifically refers to the symptom cluster that is experienced in Parkinson's disease. And this includes resting tremor, which is when your muscles involuntarily move when resting, slowness of movement, rigidity or stiffness of muscle movement, and difficulty balancing.
Could you take your mask down just while you're speaking?
Sure, I could do that for you. You can also see my face. All right. So with Parkinsonism, as I mentioned, Parkinson's disease is the one that's most well known for the symptoms of Parkinsonism, but there are other conditions as well that include symptoms like these. Some of which are medication-induced Parkinsonism and tardive dyskinesia. These ones are particularly influenced by medication use. So some medications will impact your movement ability more so than others. These conditions occur to produce symptoms of Parkinson's disease, but they are not actually Parkinson's disease. So medication-induced Parkinsonism will occur if you are on some medications that don't work with your body and produce these symptoms. Tardive dyskinesia is also similar, but the timing of symptom onset will differ. So with medication-induced Parkinsonism, symptoms arise pretty quickly, whereas with tardive dyskinesia, it may come across later on. With multiple system atrophy, progressive supranuclear palsy, and cortical basal degeneration, these are conditions which are considered atypical Parkinsonisms. So they're not actually Parkinson's disease, but they will produce symptoms that appear like Parkinson's disease with some variation of symptoms, symptom onset, and causes. In addition to the motor symptoms that we all know and associate with Parkinson's disease, there are also other symptoms as well, such as cognitive impairment and dementia, sleep disorders, impaired gut digestion, loss of smell, and urinary hesitance or urgency. So these are not as associated with Parkinson's disease, but I just want to make a note that these are also just as important and should be just as recognized. In terms of having these symptoms, experiencing Parkinsonism, it has a big impact on the way that you navigate throughout life and participate in everyday activities. So I just want to make a point about mobility disability, because Parkinsonism impacts your daily activities, the abilities that you have to engage in, say, eating or hygiene activities, in shopping, and participating in everyday outside activities. So here we see a bunch of different circles that kind of touch on the different aspects of life that we have to participate in inside the home and outside the home. So as Parkinsonism progresses, it is typically a progressive condition, aside from medication-induced Parkinsonism, which can be treated. Your ability to move around in your life space, which is what these circles refer to, is impacted greatly. So with Parkinsonism, you may begin with being able to access beyond the town, around the town, your neighborhood, but eventually this does close in and go towards outside the home and inside the home, limiting your mobility and ability to access your everyday activities and participate in life. So I want to talk more about dementia. I mentioned that cognitive impairment is a non-motor symptom of Parkinsonism. So thank you, Maddie, for talking about cognitive impairment. In addition to cognitive impairment, we have conditions like Lewy body dementia and Parkinson's disease dementia. These fall under the umbrella term of dementia. Actually, Lewy body dementia is the umbrella. Parkinson's disease dementia, dementia with Lewy bodies fall under that umbrella. These are conditions that impact cognition, and they also show symptoms of Parkinsonism. They differ primarily in timing, onset of symptoms, where Parkinson's disease dementia, you experience the Parkinsonism first, and dementia with Lewy bodies, where you experience the cognitive impairment first. But eventually, both symptoms will appear. Other things that may differ may be maybe some people in Parkinson's disease dementia may experience more hallucinations, for instance. Other aspects of cognition may be more impacted in one versus the other, like attention, language ability, ability to perceive your surroundings and whatnot. Cognitive reserve theory I also want to talk about, as this plays in part in kind of outlining what protective factors exist in affecting cognition over time. So things like higher education and occupational attainment, higher socioeconomic status, more social relationships, as Maddie mentioned, increased leisure activities. These act as protective roles to kind of mitigate the cognitive decline, to ensure that your cognitive decline from aging and disease may be less impacted when these are higher engaged in. Other things like social and psychological well-being are really important, like, again, social networks, addressing the problem of loneliness, increasing social participation and social support, ensuring that your levels of anxiety and depression are in check, not too stressed, and that your affect, your mood is also regulated. So the reason I bring all these things up is that despite all the work done looking at psychosocial roles on cognition and Parkinsonism separately, not many studies have looked at these things together in a relationship synergistically. So that's what I'm looking at for my master's thesis. Implications, as we've discussed, consider interventions, community infrastructure for adults living with Parkinsonism. And I don't want to just focus on people with Parkinson's disease because, of course, Parkinsonism impacts more than just that population. So with my study, I'm looking at 3 objectives. Objective 1 looks at all these relationships between Parkinsonism, cognition, mobility, disability, psychological well-being, and social well-being. I predict that more severe Parkinsonism and mobility disability will predict poorer cognition at the first assessment. Psychological well-being and social well-being, I predict, will indirectly affect the relationship between mobility, disability, and cognition, meaning that I hope to see the role of cognitive reserve in that relationship, that psychological and social well-being will better the results of people with Parkinsonism in terms of their cognition. Objective 2, I am looking at the role of psychological and social well-being in affecting the relationships between Parkinsonism, mobility, disability, and cognitive function across 3 time points, meaning across time. So the first objective was looking at one instance, the first assessment, whereas the second objective is looking across time longitudinally. So this one, I predict that psychological well-being and social well-being will independently and consistently reduce the effects of Parkinsonism on cognition across time, and that mobility disability will exacerbate these effects. And lastly, objective 3, I hope to investigate similarities and differences nationally across the CLSA, as well as another data set that I'm working with, which is from the Rush Alzheimer's Disease Center. So this is a study that is looking at adults age 65 plus residing in retirement communities and subsidized senior housing and retirement home facilities within Northeastern Illinois and Chicago. So this is an American study, and though it's not a national study like the CLSA, it'll give us insight still about the differences in demographics, health systems, and how those things impact adults living with Parkinsonism in an American context. So this is a very fancy graphic I have for what I'm looking at, all the factors I'm looking at. I have some mobility disability. I'll be looking at say life space mobility, which is the concentric circles that I had showed earlier, your ability to navigate within spaces around your life, as well as your activities of daily living, how well you're able to navigate with, say, hygiene, activities, shopping, cooking, etc. Parkinsonism, I'm looking at symptoms of Parkinsonism, not only just the 4 that I listed, but as well as kind of everyday activities like your ability to button up buttons, which works on fine motor function, shaky joints, balancing ability, and whatnot. Cognition, I'll be looking at aspects like memory, language, ability to manipulate information, attention, language, perceiving ability. Psychological well-being, I'll be looking at things like stress, anxiety, depression, and social well-being is going to be on social support, social networks, social participation, and loneliness. So hopefully by looking at all these things together, I'll find something unique that I can bring to the board. And I have not done any analyses yet, as this will be my summer project wrapping up my master's thesis, but hopefully I'll find something. Starting off looking at the Rush Alzheimer's Center data first. So I'll be working on that and then proceeding on to the CLSA data, but I'll still look forward to. Yeah, and this is my thank you slide. So thank you for everybody for providing your data and helping out with providing all the information that I'll be looking at over the summer, as well as the Brain Lab, my committee, Dr. Theone Paterson, who is my supervisor, Dr. Calce Smart, who's on my committee. And this project is funded by Elder Care Foundation, as well as the Canadian Graduate Scholarship. So.
Okay. Well, I'm very pleased to be here tonight with all of you, and I hope you're feeling celebrated for the work that you do in being participants in the CLSA, because we can't do this research without you. And so we are very grateful for your time and expertise. So I'm here tonight to talk about exploring depression by gender for Canadians age 45 plus during the first wave of COVID-19 using CLSA data. And I'm here as a co-author with Margaret Penning and Sean Browning from Sociology. So by way of a little bit of background literature, we know, not surprisingly, that social distancing has negative impacts on mental health. Evidence suggests that younger persons were more impacted by the pandemic, which may suggest that older adults are more resilient or stable with fewer and smaller effects in terms of their mental health. We're going to tease that out a little bit. It's a little bit of a problematic kind of statement because we also don't want to imply that all older adults are extremely, well, that are similar, really. They're quite heterogeneous, and so we want to keep that in mind as well. Lower income and education are associated with greater loneliness, and social isolation and loneliness, again, not surprising to any of us, were representative experiences in the pandemic, with women experiencing greater loneliness. In general, women's mental health is generally poorer than men's due to biological, behavioral, and psychological differences and patriarchal systems, among other causes. So the way that we approached this study was to use lenses called intersectionality, life course perspective, and then these two kind of come together in something called the intersectional life course perspective. Intersectionality, as previous speakers have talked about, recognizes that each social location, age, gender, ability, language, immigrant status, and income, for example, intersect. They're not independently affecting our health and well-being. They have intersections and interactions that shape our health and health outcomes. Life course perspective recognizes that lives are shaped from birth to death, in particular social, political, economic, and cultural contexts. That people grow together in cohorts, such as the baby boomers, the millennials, etc. That our lives are linked and interdependent and influenced over time and space. So these are things that guided our way of thinking about this research project. Now, Theone and others have already put up this diagram that suggests the data that are available in the CLSA. So for our project, we used time one, follow-up one, data from 2015 to 2018 that you provided, and also the two data sets from the COVID survey, the COVID baseline and COVID exit surveys. Our present study and objectives to use the intersectional life course approach to explore health inequalities arising from these social location factors that I mentioned, age, income, and gender. The objectives of our project were to examine whether and how age and income intersected to influence changes in depression among middle-aged and older women and men in Canada during the early part of the pandemic. And B, whether and how measures of social isolation and loneliness influence changes in depression in middle-aged and older women and men during the early pandemic. So on the far left, you see there, we've got age and income. We did our analyses by gender separately. We were able to look at this sequence of variables in the middle related to social isolation and loneliness and try to look at their influence on depression. Yeah, so highlighted are the variables in the COVID surveys that got us to that kind of address the concept of social isolation, that you live alone, spend time alone, spend time in self-quarantine, were separated from family, and had been in a public place or gathering of 50 or more versus loneliness. So those are some of the distinctions, and we could talk a lot about in the literature how we think about social isolation and loneliness, but I might just leave it there for now to look at the results. So we use the Canadian Longitudinal Study on Aging, the follow-up one data, as I mentioned, and the COVID baseline and exit surveys. We had about 24,000 adults age 45 plus in our sample. We looked at depression as the outcome variable and also self-rated mental health. I'm only going to talk about the depression analyses tonight because actually they were more robust than self-rated mental health in the results. Variables of interest again, our dependent variable was depression using the CESD-10 measure, taking loneliness out because we were looking at how loneliness mediated depression. We had the control variables of education, region of residence, urban, rural residence, immigrant status, number of chronic conditions, divided into either zero to 3 or more were the two categories. We had 16 multi-categorical variables. Can't say that. Multi-categorical variables looking at the age, income, and gender interactions. And as I mentioned, for social isolation, those were the variables that we looked at. And for loneliness, we used the UCLA loneliness shortened scale. Show you some of the results now. And I just want to highlight not the table on the left really, but the results on the right. One of them is hidden by the audio settings piece, but really to say there were increases in depression over time for women, slight decreases for men. In terms of the social location variables, women and men were significantly different in many of these. Women had higher rates of depression, 4.9 versus 4. Women were more likely to rate their self-rated mental health lower. Women were, higher proportions of women were found in the lower income categories and lower percentages in the higher income categories. Women also tended to have lower levels of education than men. There were no significant regional differences. So these are the descriptive statistics right at the beginning, comparing men and women. There were a higher percentage of male immigrants. Women had a higher number of chronic conditions, which is not surprising. We know that women tend to experience more chronic conditions, 4.5 versus 3.5 here. In terms of the social isolation variables at time 1, a higher percentage of women lived alone, almost double, 32 versus 17%. Women were more likely to be alone 8 or more hours a day and more likely to self-quarantine. Women also experienced higher separation from family, 68% versus 54%. And the two, women and men, were similarly likely to be in public spaces of 50 or more. The loneliness scale shows that women had higher levels of loneliness than men at time 1 and at time 2, but that levels of loneliness increased for both men and women over time. Oops, I'm pushing the wrong one. That's why. This is just a simple table that again shows that rates of depression were higher for women at time 2 than men, 6.1 versus 4.98. Self-rated mental health was 3.78 for women, 3.94 for men, so slightly higher for men. And the UCLA loneliness scale again shows that loneliness levels were higher for women than for men. So now I'm going to show you the linear mixed model results. And we used our reference category was 45 to 64. So we had the younger age group and the higher income group, and we compared every one of those age, gender intersections to this group. And so what we found was that in the 45 to 64 age group, low income had greater depression than the 45 to 64 high income group. At 65 to 74, high-income women and men experienced less depression. At higher education levels, there was less depression generally, but depression increased slightly over time. Chronic conditions, number of chronic conditions was associated with increased depression, but over time, levels of depression were slightly depressed as well, or tampened down. Women-specific results between the 65 to 74 and 75 plus low-income group had decreased depression over time. In terms of regional differences, comparing other groups to Ontario, in Quebec, there were lower levels of depression over time, and women in urban areas experienced increased depression. The same comparatives for men showed that, again, if you were from Quebec, living in Quebec, you had lower levels of depression than Ontario. And if you were from the prairies, for men, they had higher levels of depression. Looking at the social isolation and loneliness variables here, we were quite disappointed in a sense that the social isolation variables did not add very much to the models. Increased loneliness, however, is associated with a greater increase in depression over time for both women and men. For women only, there were no unique differences for men in terms of the social isolation variables. And for men, there was a separate difference in that those with no family to be separated from exhibited less of an increase in depression. So some of the limitations of this project, things that we're wondering about now, is the CLSA a healthier sample than average Canadians? Perhaps there's some evidence that suggests you have to participate in a lot more tests and measures, and that maybe that self-selection process results in a slightly healthier sample. We can't do fulsome gender identity analyses at this point in time. Gender identity variables have been added in follow-up too, but in the earlier waves of the data set, we're still working with male, female, and women, men binaries. The COVID-19 surveys that were part of the CLSA in 2020, baseline and COVID exit surveys, maybe they were timed too early for us to see a lot of effects in terms of loneliness. Although it was a significantly devastating time and influential time for so many of us. Social isolation measures that were in the COVID survey were not maybe the best. Could have hampered what we found as well. So different measures of social isolation or combinations of variables may be helpful in future for us. In terms of some discussion and conclusion items, an intersectional life course perspective that looks at age, income, and gender provides more information and is helpful to us to some degrees in understanding rates of depression and mental health. Depression was a more sensitive indicator of mental health than self-rated mental health in our results. What does depression among older adults not being as bad as younger persons reflect? So again, that interest in the 45 to 64 age group for men and women in lower income categories experiencing more challenging effects of the pandemic. And as I mentioned at the outset, we want to look at this quite carefully and not suggest that all people who are older than 65 or 45 to 64 are resilient, because we know that there is a lot of resilience in some individuals and there is a lot of vulnerability as well. So we need to be cautious about interpreting these results. The regional differences were interesting and hard to explain in many respects. Loneliness remains a robust predictor of depression and self-rated mental health for both women and men, however, much more than social isolation, which is a little bit more difficult to measure. And overall, a gendered analysis revealed perhaps more similarities than dissimilarities in terms of depression and mental health, even though women were more vulnerable at the outset. So future thoughts and directions. CLSA is a unique data set with many ways to continue to explore healthy aging and these relationships between social isolation and loneliness and depression. Service providers must remain vigilant, however, about understanding social isolation and loneliness and ensuring programs, supports, and services exist to combat and reduce them. And this research also highlighted that we need to be more sensitive to younger and middle-aged older adults, as well as the oldest group in difficult times. I think there's some references there, and I maybe don't even have a thank you slide, but thank you.
Hi, I thank you everyone for attending today's session. As Theone mentioned, my name is Sanjit Roy, and I'm a PhD candidate in Social Dimensions of Health at UVic, and I am also a student affiliate at the Institute for Aging and Lifelong Health at UVic. So today I'm going to share my PhD research proposal which I am studying elder abuse among Canadian veterans, and I'm using the data from CLSA. So in this talk, first I will describe briefly the importance of this study, and then I will describe how CLSA contribute to this project, and then I will briefly describe the brief overview of my study, which include research questions, theory and methods I'm going to use, and also some anticipated outcome that might come from the study. So let's talk about a little bit background. If we look at this age structure pyramid, which is constructed by census 1971 and 1921, we can see older people are increasing over time. So by 2021, almost 1 in 5 Canadians are age 65 and above. And this will continue to increase over time. And one projection showed that by 2080, almost 27% population will be 65 and above. So it's a huge population age structure transition. So this has a huge implication. As older people are going increased over time, so they are getting high research priority among the researchers and the policymakers, because we all want to make sure that as they live, they maintain a good quality of life during their lifespan. But sometimes practically it might not happen. They might undergo with different kinds of challenges. So elder abuse might be one of them. So I'm researching the elder abuse issues in my thesis. And elder abuse might have several negative consequences. So that might include at individual level and also societal level. Some important individual consequences might be the victims might have high mortality rate as they abused, and also they might face a poor physical and mental health status, and also they might have the financial loss, and overall their quality of life might be decreased. And in terms of societal consequences, there are many costs associated with these consequences. For example, the cost of hospitalization, the cost of nursing home placement, also other social and emergency service uses. So, so this is a huge consequences. So if we don't prevent the occurrence of elder abuse in future, so we should control them to reduce these consequences. And the global statistics shows that almost 0.8% to 36% populations are getting abused, but this is a huge interval. So this huge interval means different researchers use different methodology to study the issues of elder abuse. And also CLSA follow-up on data shows that almost continuing. So a CLSA follow-up study shows that almost 10% of the respondent reported any kinds of abuse issues. So this topic is called a silent epidemic because
My apologies, Sanjeet. Please do go ahead. We've cleared everything else out of the Zoom.
Okay. So the Employment and Social Development Canada mentioned the elder abuse has been considered as a silent epidemic. So because many cases go underreported, and there are many definitions of elder abuse. The most widely accepted definitions is offered by the World Health Organizations. So where they mention the older people might be abused by the trusted ones who they trust, by family members, and they might be abused in terms of physically or psychologically or sexually, and even financially. So some older adults might be neglected as well. So I'm focusing the particular group of population, the Canadian veterans group, because I just found that this group of study is very important group of population in our country. So and they have a huge contribution to their country, and almost more than 500,000 people veterans living in Canada by 2021. So, but this group of studies never been studied in terms of elder abuse issues. So that's why I'm focusing on the elder abuse issues among this group, because many literature shows that they have the high chance of being abused because of many indicators. So here's the nutshell, like why old veterans are more vulnerable to be abused compared to other non-veterans. So first of all, after the service, they might have a hard time to function properly and also make a relationship again. So that might make them a vulnerable position to be abused. And also veterans are mental and physically compromised compared to other non-veterans in terms of many health indicators. And one of them might be post-traumatic stress disorders and other mental health problems, also physical disability. There's a higher chance of having these kind of health issues. So these factors might increase the risk of abuse among this group. And next, the veterans population might have the high chance of getting disability and pensions. So they also might be abused financially too. And I did extensive literature research to find out what is happening in terms of elder abuse issues for the veterans populations. But I didn't find any article in terms of Canada, but I found very few literature, particularly focusing on the United States. Only one empirical study mentioned that there's some prevalence is higher, and also other studies recommended that. So we should focus on this particular group of people because of high chance of being abused and other risk factors. So the huge population still live in Canada and as a veterans, but the issues of elder abuse never explored for this group of people. So there's the huge knowledge gap in this context. So, and my study will be the first study in this context to fill out this knowledge gap. So I want to give my special thank you to the CLSA because I got the access their data, and also thank you all the CLSA participants for providing your valuable information. That is huge contribution to my study. The two main variables that I'm going to use, like veteran status and also abuse issues. So at CLSA baseline, veteran characteristics were asked among the participants in both comprehensive and tracking cohort. And at follow-up one, the baseline abuse issues were asked. So CLSA has his contribution for my study because I'm using their data. So now I'm going to discuss the little overview of my study, like what I am planning to gain and also how I'll do that. So three main research questions might guide this study. First of all, I want to explore the prevalence and types of elder abuse among Canadian veterans and also non-veterans, and how they are similar and different. Also, what might be the potential risk factors of elder abuse among these two groups and how they are similar or different. And finally, I also want to explore how the perceptions of healthy aging varies among these two groups. So particularly who are getting exposed to elder abuse issues. So how they are similar and how they are different. So in terms of theoretical understanding, so I'm using the concept of ecological systems perspective as a theoretical basis, because elder abuse is a complex issue. So it might happen at a different levels of the society, starting from individual level, family level, and broader societal level. So this theoretical perspective has the power to capture the entire etiology of elder abuse. So that's why I'm using this theoretical lens in this study. Since some questions will be answered using quantitative methods and also some questions will be answered using qualitative methods, so this study will be using a multi-methods approach and also using a pragmatic philosophical lens. So in terms of quantitative methods, I want to use the propensity score analysis technique, and this technique is very powerful to prepare the comparison group so that the existing results might be comparable and non-biased. And also to investigate the risk factors of elder abuse, I want to use the logistic regression approach. And to investigate the perspectives of healthy aging among veterans and non-veterans. So I will use the reflexive thematic analysis approach and will make a comparative analysis among these two groups. So as we know, CLSA has a huge sample size, which is a national study. So more than 50,000 samples are there, but so for this study, I am going to use a subsample, like because as I told before, the veterans' characteristics were asked at the baseline, and abuse issues were asked at the follow-up one. So, and abuse questions were asked who are 65 and above at that time. So, the analytic sample of this study will be the participants who completed both baseline and follow-up one, and were 65 plus at follow-up one. So for selecting variables, I just follow my literature searching findings. So I found like many indicators that might be relevant for investigating these risk factors of elder abuse. And some variables will be collected from CLSA baseline, and some variables will be collected from follow-up one. And also, obviously, all these variables are related to my theoretical understanding, so that I can explain the results in terms of my theoretical underpinning. And I told before, this is still in proposal stage. I am starting to do all analysis right now. So I'm anticipating that this study will help to understand the prevalence of elder abuse among veteran populations, and also it will also help to identify those who are at high risk among this group, and also it will be helpful to inform the development of evidence-based interventions to prevent elder abuse for older veterans populations. So obviously, I'm hoping this study will add some new knowledge in this area of research. So that is from me today. So thank you again for attending today's session. So if you have any questions, welcome.